So I'd like to thank Ranjini and Mahesh. Ranjini is not around. Yeah. So yeah, thank you for organizing this school workshop and giving us this opportunity to meet and give our presentation. Yeah, so today I'm going to talk about molecular motors and force generation by molecular motors and what they can do. Uh, so I'm somewhere here in Bhubaneswar. Okay, so a little background I mean, for the students also. I don't have any cartoon of a cell. Assume that there is a cell and there is some cell nucleus, other organelles inside, and there are two kinds of filamentous proteins, typically microtubules, which maintains the shape and stability of the cell, and there are actin filaments, typically mostly at the uh, near the cell membrane, and that these filaments and associated motor proteins that constitutes most of the cytoskeleton in a cell. So we'll just write here. So this is microtubule and this is actin filament. So we'll know what we are talking about. And there are microtubule associated proteins, typically two kinds of proteins, kinesins and dyneins. These filaments have, these are typically polar, structural polarity, so that the molecular motors, one kind of molecular motors, kinesin, work from minus to plus end, and dynein works from plus to minus end. And this is a family of motor proteins. Various kinds of motor proteins do that. And by doing that, so there is what they can do is that they can transport cargo from one part of the cell to other. The cells are typically several microns in size, and I'll be also talking about something called uh, neural cells, which has typically a cell body where this nucleus, etc., structures are there, and long appendages called axons. And in one part of the talk, I'll be talking about transport of molecular motors, kinesin kind of molecular motors, particularly kinesin 3, uh, moving cargo from the cell body towards the cell periphery, say long appendages. So those cartoons will help us. Uh, so they can transport cargo. There is this small animation, hopefully it works. Yeah. So I mean, it looks like there is this small molecule which holds a cargo. So there is one domain by which it holds a cargo. With another domain, it walks. So typically, this walking domain is called tail, and by which it holds head, and by which it holds the cargo is called tail. So these two domains play some important role because you have to, so first you have to walk and also you have to hold cargo to generate reliable transport. So this can generate transport. So these are these two kinds of filaments I showed, microtubule and actin filaments. Apart from that, if molecular motors cross-link between two filaments, then and try to walk. So cross-link two filaments, you can imagine this kind of structure that, let's say, I have these molecular motors. Somehow this one is plus and that's minus. And there is an assembly of molecular motors, some of which these guys are trying to walk towards this plus end, these guys are trying to walk towards the minus end. If such a situation arises, that will generate a shear stress inside a cell. But of course, if they are parallel, then this complex itself can move because both sides will move in the same direction. So they can slide or they can generate shear stress. So these are the two things which will look at the impact of these two properties of molecular motors. Okay. So as I mentioned, cytoskeleton is made up with filaments. crosslinkers. So crosslinkers I did not mention. There could be also passive crosslinkers, or which does not move but just crosslink different parts of the filament, and thereby they can give stability to these filament structures. But typically in all these systems, if they crosslink and stay there for some time, and under stress they can detach and then reattach. So over short time scales, this kind of crosslinks can give rise to a solid-like behavior, and at a longer time scale, this will generate some viscoelastic properties. Okay, so that's kind of introduction for students whoever are uninitiated. Uh, here, these two experimental uh, plots are showing that this fluorescent proteins are tagged actin cytoskeleton. You can see that actins are mostly on the cell membrane, near the cell membrane. These blue parts are the nucleus, so tagging DNA. And here you see again these two nucleus and see microtubules. Microtubules are mostly in the bulk. So what I tried to show here on this cartoon, that this is mostly on the bulk and actin filaments are mostly on the cell membrane. So the generating stress and beyond transport, generating stress, they can do various things. For example, yeah. So this cell division, you see this here. This green stuffs are the microtubules, which are generating this microtubule uh, structures are pulling the molecule, pulling the chromosome into dividing cells. So after chromosomal replication, they attach in this spindle structure, and then they pull and break the sister chromatid threads and take them to two different cells. And here I show this neural cells. So this is the cell body, and this is the axon, and this is the end of this cell. So terminus, where you see this growth cone. And so two things I'll be talking about if time permits. First is that we'll be talking about transport along this cell, looking at properties of molecular motors along this axon. And if time permits, I'll also go to membrane fluctuation. So this growth cone is almost like a two-dimensional membrane having lots of actin filaments, that's the red stuffs, and generates huge amount of fluctuations, that it helps for the cell to identify cell addition molecules coming from other cells. By that, it can figure out whether it should go towards another cell or go away from other kinds of cells, and helping them to develop neuronal structure network. This in the left red is the chromosome, and each figure has different color codes. I'm sorry for that, taken from different sources. Red there is the chromosome, and blue, the green is the microtubule. Okay. So then let me come to the outline of my talk. My plan is to first talk about regulation of cargo transport along axons, and in the second part, talking about uh, dynamics of semi-flexible filaments under force developed by the molecular motors, and dynamics of membranes under force developed by molecular motors and membrane associated proteins. Okay, so this ongoing work. The work is mainly done by Vidur in Sandha's lab, and Amir, along with Amitavo, we developed the theory, and they also did all the data analysis. Amir did all the data analysis. So this cartoon, what they are doing is that they are looking at C. elegans, so living C. elegans, which has precisely 302 neurons, and all the neuronal types are precisely known in C. elegans. And in these experiments, they're looking at these mechanosensory neural types. Mechanosensory neurons means that if I'm hurt, how I feel that, heat, etc., how I feel that. So those are mechanosensory neurons. Uh, so what this cartoon shows is that this outer line is the outline of the C. elegans, and inside this green part, so this little blob stands for the cell nucleus, where you have the cell body, and then the rest of the part is the axon, and then there are these connection between axons which are called synapses. So I'm simplifying this cartoon further, another cartoon. So in this cartoon I'm showing this as the cell body, and this part of the axon is called process, and that's the distal tip, and it can have several connection with other neurons which are developed via synapses. So on this system, we are looking at particularly the kinesin 3 kind of proteins which walk towards the plus end of microtubules. And in this system, it happens that the microtubules are aligned uh, in this manner. So plus end is here, minus end is there. So of course, this is not a single microtubule, several microtubules uh, stacking one after other, generating this connection. And then these molecular motors are supposed to go from the cell body towards the distal tip. So of course, if you look at dynein motors, they will be walking in the other direction. All kinds of things are required. But in these experiments, typically what is done is that you fluorescently label this specific kind of kinesin molecule. I'll give the name. And that molecule, if you tag them, then you see transport due to that molecule alone. So this is a generic statement that the axonal transport is essential, of course, for the proper functioning of the cell. And you have to imagine also that most of the proteins are generated here. They needs to be transported to other parts of the cell, so that even the cell membrane growth, and when the cell is growing and after it's grown and it maintains a structure, one needs to also, to maintain that structure itself, one needs to keep supplying material. So generic questions are, what is the mechanism of cargo transport? And cargo means that, so typically what happens is that uh, cargo, all kinds of proteins, whatever is there, is taken inside a vesicle, and then the vesicle is transported by the molecular motors. So that's what we are showing this in this cartoon, that vesicle being transported. So how is active cargo transport chemically regulated? That's the second question. So these are generic questions people ask in this field. It cannot go back, but it's stochastic. So I mean, whatever in the cartoon is shown is that it's like deterministic. So not deterministic, it's stochastic. If you follow the trajectory locally, at least for certain kinds of kinesin molecules in lab, people have seen that it can do a little bit of backstep, but it pauses a lot more than taking backsteps. But backstep probability is extremely small. It's typically in the front step. So roughly, it's moving in the same direction. In the presence of noise. Okay, I forgot to mention that very important aspect.
Thanks.
Strong molecular noise. Okay, so several kinds of modification one can imagine that one can do on top of these molecular motors and ask how they impact. So one specific type that Sandha's group is interested is in ubiquitin pathway. So ubiquitin proteasome pathway, this system degrades intracellular proteins. So, and this degradation is important because protein is generated and it can change after doing some work, it may change its shape, and once this is not useful, you want to degrade it and you want to get into the molecular basis of this so that you can generate more other proteins, whatever you require. So you don't want to waste the waste material. And several neurodegenerative disorders, all these guys, Alzheimer's, Huntington's, Parkinson's, they are characterized by aggregates in the brain if proteins fail to degrade. So in that sense, these guys are important. So then the question is asked is that what is the role of ubiquitin to transport in ubiquitination pathway? Ubiquitination happens on top of most of these proteins. So what happens is that a little detail that ubiquitin proteasome pathway, ubiquitin goes and gives a tag. So multiple ubiquitination gives a tag to a protein and telling the protein that this protein needs to be degraded, and that will ultimately proteasome will degrade that. So that ubiquitination, that ubiquitin structure is there in this kinesin molecules as well, the molecular motors. And one asks the question that if you perturb that, what happens? Okay, so what happens? First result is this, that if you look at wild type without any perturbation, if you look at cargo accumulation, cargo carried by this specific kinesin 3 kind of molecular motors, cargo accumulation, you see that there is a little haze, little white region here near the cell nucleus, and then mostly there is a high density white regions near the distal tip. So there is cargo accumulation near the distal tip, cargo is going there, but if you modify this ubiquitin pathway, in the sense degrade the ubiquitin pathway, you don't allow the ubiquitination happen properly in various ways. So if you do that, what you see is that there is more accumulation near the distal tip. So that's the first result we see. Uh, so question is that one sees this accumulation in the distal tip, uh, how it happens. So another question one can ask that whether it's really like transport of the molecules, the picture I'm saying that this is being transported, if that picture is correct, and getting accumulated here, if that picture is correct, and if I uh, cut this action somewhere, the accumulation will happen here, so not on the other side. So that's what exactly the experimentalist did, that if you do a laser ablation somewhere in between, you see this accumulation of this cargo in that ablation site, and that you can quantify how fast it accumulates. So this rate of accumulation one can calculate and use, of course, just see this accumulation happening there. So this confirms this idea that there is this what is called anterograde flow going from the cell body to the cell terminus, and this increase in anterograde active transport in the mutants. This active transport, so these are these mutants. This is the original wild type, and these are these mutants, and you see this greater accumulation at that place. So this is the question that with suppressed ubiquitination in mutants, we see this accumulation, but is it due to what? Is it due to better transport, better cargo binding, or both? So I sort of showed in the beginning that there are these two domains of the molecular motors. One domain works, so that's transport. Another domain holds the cargo, so that better cargo binding. If I don't hold the cargo, I cannot take the cargo there. So what is happening? Okay, so this is just uploaded few days back. If you are interested, you can have a look at it. This we are still working on. Maybe next week, next month, this will be uploaded somewhere. Okay, so this is a work in progress. Of course, we know certain results now. So what is being done here is that you can look at this molecular motors. So if you tag the molecular motors with fluorescent label, Looking at high intensity, you know that there is more accumulation of molecular motors. Low intensity means lower number of molecular motors. So you can look at these intensities and see how they move as a function of time. So here on these plots, this is the length axis, that's the time axis. Length means going from this point to that point. So these chymographs show you that some of these lines you see that they are horizontal, means that things are stuck where they were, they are stuck. So something is happening. But apart from that, there are several trajectories which goes in this direction, several other trajectories which has non-zero slope. So whatever has non-zero slope, they are plotted from these slides, they are extracted here. So you see most of them are going in the anterograde direction. Some of them is going in the wrong direction, retrograde direction, the opposite direction. So one possibility is that these molecular motors can still remain attached. Let's say kinesin is attached here, but dynein is carrying the cargo. So that's possible, and then you'll be seeing that some of the kinesin is going in the wrong direction if they are not attached. So to transport, they need to attach to both cargo and the railway track. So otherwise they cannot transport. So okay, from this, what one can extract is that from these trajectories, one can extract what is the velocity of individual puncta. So this run length and velocities are calculated. What we see is that velocity, so these are distribution of velocities. So along this direction, you imagine this is the distribution function. And these velocities have number between like roughly around 1 micron per second. So that number sort of corresponds to other kinds of kinesin molecules and uh, uh, even in vitro experiments. So next comes, so this we are seeing that this transporting of each individual molecular motors when they are in the attached state. The second question is that cargo binding statistics. So what happens if one changes this so under mutation, so this is in the wild type, what is the, so what you are asking is that what is the number of molecular motors attached to a cargo and distribution of that. So you see that this distribution, the peak shifts towards larger values under these mutations in which the ubiquitin pathway, so specifically E3 ligase is perturbed. So you have better cargo binding, but this distribution can we understand? So that's the first question. Okay, I'll just show it the full thing. So one simple way to try to understand that is that if you write down a master equation for you look at a specific cargo, ask a question how many molecular motors are attached to the cargo at a given instance, and ask a question that what it can do is that one molecular motor can detach or another new molecular motor can attach. So depending on that, if I give this probability, P of nt means that n number of molecular motors are attached to a cargo at a given instance. So it can gain, there could be gain terms because n minus 1 number of, so cargo with n minus 1 molecular motor can get a new motor, and that's the gain term. Cargo with n plus 1 number of molecular motors can lose one. That will also lead to a gain term. And similarly, there will be loss terms. So this cartoon probably will be easier to see that if you start from here, you see that there are like 4 motors are attached here. If I ask a question that I jump there, lose one motor, that's why I'm calling this probability that u minus u minus is the probability with n minus 1 molecular motors. So that's the rate by which it can lose, and this is the rate by which it can gain. So this loss and gain rates you put together and get your probability distribution, and then there is a boundary condition. Boundary condition is slightly different because there is this cargo floating around, molecular motors floating around. The first motor, if it binds to the crosslinks between the cargo and the filament, that's given by this guy J plus. So once that happens, then the cargo comes in the vicinity of the filament, and then for other molecular motors is sort of equivalent thing. But otherwise, this is a diffusion limited process that this guy needs to diffuse, come close to the filament, and only then a molecular motor can start to attach.
So you're saying that these kinesin motors are already on the microtubule, which are working without any cargo.
No, so I'm saying that picture is this, that maybe these are motors and this is your cargo and they are not anywhere nearby, then nothing can attach. So you have to give, so this is a diffusion limited process. This guy diffuses around, and if it comes close to the filament, then molecular motors are floating nearby. If they cross link, so then it binds the first time. So once the first cargo is bound, uh, first molecular motor binds to the cargo, then the cargo is sort of in the vicinity of the molecular motors. So this other probabilities doesn't involve this diffusion process. Other probabilities are just stochastically binding to this. So I'm not distinguishing between and whether it's bound there, whether that's bound there. So what I'm saying is a cross-linking probability. So unless it's cross-linked, it cannot transport. So the full cross-linking probability. Okay. From this one can go to the steady state, and this simply steady state probability is property is this, and then for any other states, this like pairwise balance, and then one can write down the probability distribution in this fashion. So this is exact, but doesn't help us much to analyze the experiment. So we want to we need to know what is this rates of attachment and detachment of the uh, molecular motors with the cross-linking molecular motors with the filament. So detachment rate is simple, that this has to be linear in the number of molecular motors. Unless you have a molecular motor, it cannot detach. So at n equal to 0, detachment rate should be 0. Attachment rate could be, the simplest assumption could be this is a constant, but with a constant rate it attaches. So you can do that, or you can keep this other term b plus n, which I wrote as cooperative binding. So if I write down this expression, oops, sorry. If I write down this expression, then it becomes sort of clear that if, let's say, if you assume that b plus n is absent, so you can put this expression and then the previous expression will give rise to this structure. So this structure tells that this distribution grows as n to the power gamma and then decays as exponentially with n. But with this prefactor mu. And look at this prefactor mu is 1 minus b plus by beta. So if I said b plus equal to 0, then this mu is equal to 1. So if that is the case, if mu is equal to 1, then this distribution will have exactly the same structure for all these mutants. But from experiments, we have seen that this distribution changes as you do these mutations. And that tells us that this b plus by beta is necessary. Otherwise, you will just get a function n to the power gamma e to the power minus mu, e to the minus n without mu.
Number of motors present on the cargo or number of engaged motors?
Number of engaged motors.
Okay. And from GFP, you are estimating the total number of motors, right? From GFP
So
intensity.
GFP, we are looking at only those puncta which are moving, motile puncta.
Okay. All right.
Yeah.
But
True that we can only see total number, we cannot see all. So we are saying that this is in the moving puncta, this should be proportional. And we are, of course, what we are seeing is intensity profile, not number. So if you say that the intensity profile is proportional, intensity is proportional to the number, then that's this picture. Okay. So yes.
So a lot of these look like log normal distributions. Is there a reason why this cargo looks and not a normal distribution?
The reason is that, okay, so if you think about this dynamics, the dynamics tells you the reason, right? So there will be attachment and detachment processes, and that leads to the total number of cargo at a moment, total number of molecular motors you have on the cargo at a moment is dependent on this attachment detachment processes, and that's not a Gaussian process.
So what determines? So I
What determines
yeah.
is
So
what
what
determines
makes it go towards other distributions which are not normal? So
which are not.
which are not normal. So why is it that a normal distribution is not what you see in such processes?
Why it's not a Gaussian distribution? Why?
That's right. Why
Yeah.
is
So
it that you see a log normal distribution rather than?
no. So I mean, I won't call it log normal distribution because I got this distribution, right? So this is not really the log normal form.
Okay.
Yeah.
So sure, I thought it looked like a log normal
Yeah.
distribution more or less, but
I mean,
fine, you fit
structurally.
into
So
some
you
curve and
can
you have
say that
extracted the
it looks also like Poissonian. So it
sure.
looks like many other known distributions. But this is what
You fit it to some form of an equation. But is there a rationale why you picked this, or it just came out of the fitting?
No, that's the rationale, right? So rationale is direct, that you have this cargo with which molecular motors can attach and detach. That's the dynamics, which will give rise to the certain number of molecular motors in a steady state, right? That's the dynamics. So we just taken that dynamics, did not assume anything else. So that will lead to a steady state. Sorry?
Why is it asymmetric is the question.
Yeah, so that asymmetric part that's coming because of the attachment rate. So what is giving rise to this? That there is an attachment rate which gives rise to this growth, and then even the decay itself is not given only in terms of detachment. Decay itself is given by this cooperative binding B plus and this detachment. So that's what dictates these properties, and then if you take this together, what you see is that for all these mutants, this cooperative binding rate is least for the wild type and maximum for this mutant. So this cooperative binding rate itself is changing, but nothing else. Others remains almost the same. All other parameters remains almost the same. So this is the cooperative binding rate, which itself plays the major. So which stays on the ubiquitination pathway. So wherever you are modifying this ubiquitin region on the molecular motor, that region is coupled to the cargo binding region. Okay, so that's getting modified. Okay, so what we showed here is that this can increase cooperative cargo binding and the modification impacts the cargo binding domain. Then comes the other question, that what about the transport? So what about how does one write down a equation for the transport? What in the experiment people can see is that this is the intensity profile of these molecular motors everywhere. So that gives a density profile. So you don't really see how many molecular motors, so in general experiment, you don't really see how many molecular motors are attached, how many are detached. Whatever is present, they will fluoresce and you will see them together. So for that, if one tries to develop a theory for that, so there are bound fraction of molecular motors, there are unbound fraction of molecular motors. One can approximate that the bound fraction of molecular motors are the only guys which transports, so that has this drift term, and can imagine that the diffusion there is less, and just ignore that diffusion with respect to the drift for the bound fraction. And for the unbound fraction, it can only diffuse, it cannot drift. So keep the diffusion term there. And then there is turnover between the bound fraction and unbound fraction. So what you can do is that you can write it down in terms of so the total density and the difference in this density. And if you do a adiabatic elimination for the difference in density, which turns out to be a faster degree of freedom, so you can write down a equation for this total density here alone. And then in this, what happens is that this is a renormalized velocity, renormalized by the processivity omega, so which gives you the fraction of time the molecular motors stays in the crosslink state. And the diffusivity is also renormalized by this processivity. But if, so you see that if I stay lot more with in the attached state, then this velocity will be like the velocity of individual molecular motors when they are moving in the attached state. But if mostly it's in the detached state, this effective velocity will be less. On the other hand, if it's mostly in the detached state, the effective diffusivity will be more because detached fraction diffuses more. Rho B and rho U, you add them together and then you do this adiabatic elimination. That will give you a relationship between K off and K on in terms of rho B and rho U. And then you plug them in. And this equation, then what you'll see is that this changes to a new drift, which depends on this ratio on and off. And this on and off ratio is giving rise to this processivity. And that also changes diffusivity. Yeah. Okay.
Question.
So,
So,
yes.
in the previous slide, this unbound density, it contains both the proteins which are not connected to both, right?
Yeah.
So
Yeah.
if a protein is only connected to the cargo,
Will not help.
yeah, but
So that will be diffusing with the cargo. In general, you should then change the diffusivity. So lot more complexity one can imagine.
but in the
Is
last slide you showed that if for a one cargo there could be many motors connected.
mm-hmm.
In that case, the unbounded one also will go with.
So this is the idea is that this is the density profile. The density profile, you now look at a local density without distinguishing between individual motors. Earlier I was asking a question that I look at a specific cargo, ask a question how many molecular motors are attached to that cargo. So that was a more local question. Now I'm asking a more coarse-grained question in which I'm asking that what is the transport of this density profile? And in that local density profile, some part will be detached, some detached means that will be diffusing, some other parts will be drifting, right? So now you can take these two parts and they together gives rise to this, the total stuff, and try to look at the dynamics of this total stuff. But this is not all, I'm just trying to develop this effective equation. So this says that if there are these molecular motors which are doing this drift diffusion, that will give rise to this effective equation. So once you have this effective equation, two more things you need to add. So one is there is a source that these molecular motors are, all the proteins mostly, they are being produced near the cell body. So that source is near this x equal to 0. So this is plotted in the other way. So this is x equal to 0, this is x equal to L. And then these molecular motors can also degrade along the way. So that's this degradation rate. So together that describes the full dynamics of this density profile. And now if you take this profile and go to the steady state, you can figure out what is the steady state distribution of this. And this steady state distribution involves several length scales. One length scale comes from the ratio of diffusivity and drift. Other length scale comes from the ratio of diffusivity and the degradation rate. And then there is this composite length scale that appears here. Composite of both of them. Okay, so that's a distribution function, steady state distribution function. So one can go and look at the steady state distribution over the whole cell and try to extract some quantities from there. And you see that some quantities in the sense that uh, in this expression there are these quantities drift, diffusion, which are the transport quantities. So you would like to extract the transport quantities. Okay, so first thing, that if you want to extract all the quantities from one graph, this will be extremely bad. And from data, you cannot extract everything from one set of measurement. So what we try to do is that do a local FRAP experiment. So FRAP is that locally you shine laser, this fluorescence recovery after photo bleaching. So if you do that, you can extract the dynamics locally. So there we do a local flat approximation of this equation, and then one can write down the corresponding solution of that equation. So that solution comes out in terms of error functions, and then you try to fit that. So you plot this solution along with the experimental data of the FRAP, and you see that, and by that you can fit for the diffusion constant. So we are extracting the diffusion constant number one from here, and also like if you integrate it over, you can look at the evolution of the total number in a window, and that FRAP window, and that also has encoded diffusivity. So in these two different manners, we extract the diffusivities of these molecular motors. So news is that one can do this, but wild type and all these mutants we looked at, all of them show diffusivities of the same ballpark. So they are not too different from each other. So diffusivity is not changing much. And that's sort of understandable because diffusivity is mostly due to, so how it's understandable, I'll come back slightly later. But let me tell you what it is, that in a separate experiment, we can also figure out what is the effective drift, and effective drift and diffusivity depends on this processivity. And we see that processivity is extremely low. So diffusivity is mostly controlled by the free diffusion. So doing mutations will not modify that. So that's kind of understandable post facto. Yeah. Yeah.
Here, you have only diffusivity is the fitting parameter.
So others are also, you can fit all others. So what we are not trying to extract those numbers from this graph. So what you can fit from is the diffusivity, the degradation rate gamma. And since this is happening very fast and we know the degradation happens over a very long time scale, we are holding that to be, so sort of like gamma is 0 within that time scale.
Time does not change, right?
Diffusivity with time doesn't change. So we are fitting it. So you can fit this function with a single diffusivity. The time is changing here. So yeah, but if you fit that, then you'll extract the diffusivity from this. Yes. Drift. Yeah, there is drift sitting here. So I'll come back to the issue of drift. There is a tricky issue with the drift. So that I'll come back at the later stage, because we are really confused with the drift coming out from this guy. So I'll show you why we are getting confused and decided not to confuse you at this stage. Okay. Yes. What is? P of x. Oh, phi of xt. So that's like local density profile, density as a function of time. But what we measure is intensity, right? So there is a factor. So intensity, we are assuming that intensity of coming out of the molecular motors is equivalent to the density of molecular motors present there.
So you need this because this is what you can see in experiments.
This is what the dots are what is seen from the experiments, and the line is this equation. Okay. So the conclusion is that diffusivities remain unaltered and the number is like several microns square per second, 4 5 microns square per second. Okay, so then finally what we do is that we look at the intensity profile along this axon starting from the cell bodies to the cell terminus. So this is around 300 microns in length. So over that steady state intensity profile, all of them shows this exponential structure. So somewhat simple to understand that this is being generated here, being carried there, and then there is a boundary. So it's boundary accumulation. And of course, there is production and degradation, which maintains the overall rough number of the total number of molecular motors throughout the system. And then this is a flux which is getting blocked at this end. As a result, you see this exponential growth. So, but different exponential growths, you take our steady state distribution and fit with them. So all of them fits well. So I said that degradation rate is so inverse of degradation rate, the time scale is too long, several hours. So don't worry about these numbers. So we just kept it for its sake. What happens is that I said the diffusivity is almost unchanged between all the mutants. If you look at velocity, you see that in two mutants, velocity goes down by 1 order of magnitude, but this is an effective velocity. Remember that the velocity from individual motor proteins that we measured earlier was like 1 micron per second, and this guy is like 0.01 micron per second. So this tells us that the processivity is extremely low. So that's between 0.01 and 0.001. This degradation rate is about 1 per hour, which agrees with other measurements. And this also gives you the production rate of the molecular motors. But main issue is that only drift changes a little bit. Diffusion does not change. And of course, this remains outside the scope because it is too long time scale. And but still, we saw this accumulation of molecular motors at this accumulation of cargo at this end. So the accumulation of cargo is happening. Earlier, I said that cooperative binding is increasing. So that's the main factor in accumulation of cargo. So that leads to it. So now I'll come back to the question that Guru asked. So what was the issue with the drift? So what happens now is that, so initially when we are doing this FRAP, so we thought that locally, up to that moment, the intensity profile was measured only locally, and it looked like pretty flat intensity profile. So we thought that it's a flat distribution, and you can then do a FRAP and try to figure out if there is a drift from right to left, you'll get the drift. If there is a drift from left to right, you will get that drift. But what happens is that profile itself is not flat. Profile is this exponential profile. Now you can imagine that if you are doing the FRAP here, so wherever you do the FRAP, after the FRAP, this line will like to go back to the steady state. So if I am doing, let's say, a FRAP here, going towards the steady state will generate an effective drift towards left. So one can start to get from the FRAP result, you will be concluding that I am getting retrograde flow. But that's not retrograde flow. That effective drift is coming due to this background profile. So I'll run this then. You see that this is that local FRAP. So this guy is trying to go towards that. And as a result, this minimum is shifting, although a little bit, shifting towards left, although the molecular motors are going all towards right. So this is the crucial issue while doing a FRAP. If we don't know the steady state profile in the background, that can generate such a wrong conclusion. So we are getting stuck to that conclusion for a really long time before we figured out that steady state profile should be exponential, and this measurement showed us that. Okay. Yeah, so what we found is that the cooperative binding can, that's essential in this generation of excess cargo accumulation at the end of the neural tip. Ubiquitination play a crucial role in regulating this cooperative binding domain, but processivity can decrease a little bit, but otherwise rest of the transport properties remains unaltered under this ubiquitination. So the take-home message is that ubiquitination hampers the cargo binding domain, not the motor domains of the molecular motors. Okay, so I don't have much time. So what I'll quickly do is that I said that filaments driven by molecular motors, what are their structures? So I can just quickly show that. So this is the work done by Chitrak and Amir, who Amir was a PhD student in IOP, and Chitrak is still a student there. And Nisha did this with Abhishek in ISAR Mohali. So what we are thinking about now is that these molecular motors, they are not inside a cell. You take it out, and then these molecular motors are, so this is a molecular motor assay setup in which these molecular motors are bound head down. So what is happening is that so this is this substrate, on this substrate, the molecular motors are attached here, and this is the domain which you would like to work on top of a filament. And if it works on the filament in a certain direction, the filament will be pushed in the opposite direction. So that will generate a drive on the filament, and that drive will be typically locally tangential to the filament. So if one takes that, so there are several experiments, many body experiments, single particle, single filament experiments, which people have done over the years. I'll not get into the details. So main issue is that these filaments are, the cytoskeletal filaments are typically semi-flexible filaments. They have a bending rigidity. So we incorporate that bending rigidity, and then there is self-avoidance between different parts of this filament, and then there are these molecular motors which will drive the filament locally tangentially. So, and for that there are rules that molecular motors, if they are free, then they will maybe will be generating a velocity like 1 micron per second that we are seeing in the previous part. And then if there is a load, the velocity, the transport velocity will decrease, and the decrease is like this. So this is some experiment and fitted with this equation. And one can extract these numbers from this equation and then take them together. So what is happening is this molecular motors can, so now we are doing a full-fledged simulation in which molecular motors can bind, and once they are bound, they will walk, and if they stretch a lot, they will unbind. And of course, there is also a intrinsic timescales of unbinding. So there is a binding, unbinding timescale, and unbinding rate increases if it stretches a lot, and that will generate force on the filament. So that will finally lead to this, the most interesting instability that it generates on top of the filament is that filament gets into the spiral structures. So these are the spiral structures, and they stays in the spiral structure, and depending on what parameter values, what activity values you are, so they can stay in the spiral, come out of the spiral. So I think this movie was showing how it comes out of the spiral. So initially it was in a spiral, then if you wait enough, they'll see that it folded back and it's unspiraling the spiral. So both those things happen. So it can go into spiral, come out of the spiral. So there are these dynamics, and we can extract. So we extracted a phase diagram in which, depending on the attachment detachment rate and the activity, which one can control by the ATP concentration. So one can get these different states which are like open chain structure. Then there are spiral filament, predominantly spiral filament, and then if you increase activity further, this can become again predominantly open chain in presence of spiral. So where it spends little amount of time in the spiral state. So then we did several response function calculations. So I'll not get into this. There is not enough time. So quickly, what I'll try to convince you with is that, yeah. So quickly, what I'll try to convince you with is that these things, then we got this nice, interesting dynamics. Then a question one can ask is that, what is the effective active filament picture of this semi-flexible filament driven by molecular motors? So can I write down it as some kind of a effective active filament? So to simplify that, to take all the impacts of the molecular motors in terms of certain mean force and force fluctuations, if that's possible. So we're trying to do that, and what we saw is that mean force we can sort of we get the qualitative behavior of the mean force, not the quantitative behavior. The force correlation function we can capture exactly with the correlation time. the fluctuation strength is something that again we miss out a little. So what is happening here is that, so one final point, what is happening here is that if you have multiple molecular motors on a rigid segment of a filament, locally it's rigid. So if multiple molecular motors are working there, so what happens is that one molecular motor can, the force generation by one molecular motor can generate an effective load to other molecular motors which are attached to that filament. So via the filament, one molecular motor's force can act like a load force to the other molecular motors. So it's a filament mediated self load, so which generates a little lack of cooperativity, which generates this mismatch of the mean fields, generates this initial mismatch of the mean field approximation of the force generation, because in this mean field approximation we did not incorporate this self load that this competition between molecular motors generate. So once you take that into account, you can estimate it exactly. That will that incorporation of self load will be enough to describe this mean force. So yeah, if you are interested, you can have a look at this thing. I could not get into this detail, so I'll just jump to this slide. So this the people who worked, the PhD student who contributed in this work are all these people, and then Amitabh from IIT Bombay, Sandhya from TIFR. So the last part I did not talk about, Shashi from BITS and Abhishek from ISAR Mohali. And I'd like to thank these funding agencies. Thank you for your time.